Hi, welcome to the Bioinformatics chat. Today I'm speaking with Alexey Sergushichev. Alexey is assistant professor at ITMO and head of Bioinformatics laboratory at ITMO. And also Ekaterina Vyahhi. Ekaterina is COO at the Bioinformatics Institute. Guys, welcome to the podcast.
Hello, Roman.
Hi.
So the main reason I invited you today is because you are the organizers of the Bioinformatics contest, which ended just recently. It's interesting to talk to you about the problems and also about the organization of this event. Before we get into that, I want to find out more about your background. So, Alexey, how did you get into bioinformatics and what has your path been like?
I am graduated basically from my university. So it's started from informatics, from Olympiads in informatics in Russia. I believe you are also trained in informatics and took part in this contest. So, and one of the best universities in Russia in computer science is ITMO, and also known for world champions in ACM, ICPC, and so on. So I applied there, studied there for 6 years, basically my bachelor and master degree.
So when you applied, were you already interested in biology or just in computer science?
No, I just was informatics. So, and I never expected in high school to do any biology related anything. So I was interested in programming, so competitions and so on. So I thought that I would be a programmer and went to ITMO because, that's where a lot of good programmers are studying, and a lot of my friends went there. And around third or fourth year, there was some idea to our head of our department. So it's department, it's computer technology department, was the one that basically teaches, trains these champions in ACM, ICPC. But the problem was that a lot of people, they went working in Google and Facebook. If there was there was a Facebook there then. So there was the idea that, probably it's interesting to get these good programmers to do some, science. So our heads, Anatoly Shaluta and Vladimir Parfenov, invited Yegor Prokharchuk, which is he's doing bioinformatics in Russia. So he gave a talk about genome assembly and so on. So a few guys, first without me, started to work on genome assembly problem, which was stated pretty computationally or algorithmic problem. So basically as a normal contest. Yeah. So I'm starting from that. It's a year after I joined, and basically two of my teammates, because in ACM ICPC there are 3 people usually in a team, and my teammates, also group mates, also joined me, and we did some genome assembly algorithm development. And around the end of the my bachelor project and master project were about bioinformatics, algorithmic bioinformatics, and then at the end of the master year, I met Maxim Artemov, who is a professor of pathology and immunology in Washington University in St. Louis, who's he's an immunologist now, and there it continued to be from simple kind of simple algorithmic bioinformatics to more systems biology and stuff like that. So the real biology came there. And basically I defended my PhD under his supervisorship, and here am I still at ITMO, heading a bioinformatics laboratory.
Pretty cool. Ekaterina, what about you? What led you into bioinformatics?
Well, my initial training is not connected to bioinformatics. I studied higher education, science and administration in University of Oslo. And pretty much before that, I was engaged with various projects in the education sphere. And for my master thesis, I was doing research on universities and industry connections in St. Petersburg. And I end up interviewing a lot of people connected with IT and bioinformatics. And I thought that that was really interesting and cool field to be engaged in. So I joined the group of organizers of bioinformatics courses at St. Petersburg Academic University, which further we developed into Bioinformatics Institute to experiment more with the programs and projects like this contest.
When did you get this idea to organize a contest? How did that come about?
Well, I guess it happened like 2-3 years ago. We had this idea of having something similar to programming contests in the field of bioinformatics. But it's set as an idea for a long time, and then we met Alexey and his team from Atmo University, and I guess it started actively developing from that point.
Yeah, so like one time, Katya and Koля actually said that, well, we have an idea to do a bioinformatics contest, as similar to other contests like programming. So basically it was a nice idea. Well, we said, okay, great, let's do this. But the problem was that it's hard to do problems for like bioinformatics contest, because you need a lot of bioinformatician. So we tried to ask our peers for the problems, but it's also like took like a year or something to get a set of ideas for problems for the first contest.
Yeah, because there's no prior art, right? Are you guys aware of any other such bioinformatics competitions, or is this like literally the first one, or I guess the second one? The first one was last year in the history of humanity.
Well, like first of all, there is Rosalind, which is not a contest, but it's a platform with a lot of bioinformatics like problems, like algorithmic problems. There are also several other like competitions, like dream challenges, where they have not really bioinformatics competition, but it's maybe more like computational biology. And so there are a couple of them. And like recently, I believe the first one was a year ago. It's a Topcoder precision medicine challenge, like basically a set of challenges also in algorithms, bioinformatics. So there are a couple of similar, but they are not called contests, and they are very specific. Like basically, each one of them is one particular problem that they want to solve. I believe there are and in normal contest in programming, there are like usually several problems that people should know like a lot of things to get to the first place.
Yes, and in the beginning we were thinking a lot about basic questions like the duration of the contest. Is it real to solve a bioinformatics problem in 24 hours, so we need a week? Or we want to do it more like traditional programming contests, to be it more, like active, like you have 24 hours and you need to do it fast, like in a hackathon style. So there were lots of discussions on that.
Yeah, basically one of the things that we tried to replicate, maybe, or like to repeat, was Challenge 24, which has interesting problems, and it had to be solved in 24 hours. So we decided that it should be a good format for us too. So their problems are not domain specific, but the format is kind of clicked with us. So we decided, 24 hours is great because we can have online competition for all the world, like without time zones problem and so on. And for qualification, we decided that we don't want to have a lot of pressure for participants. So that like a week should be enough to start working and start solving something.
Yeah, so for those who are not aware, the bioinformatics contest both last year and this year consisted of two parts. The first part is the qualification round, which lasted a week. And the second part is the actual competition, which lasts 24 hours. So what was the reasoning behind splitting this into two parts? What's the motivation for the qualification round? Why not just have the actual final round?
Well, there are two parts. First is, at least like the first time, we were not... So one of the reasons why the bioinformatics contest happens is to test Stepic's platform. So we were not sure like how it will respond to a lot of participants. So we decided that, well, in the finals, we want to limit the amount of participants probably. And also the other part is more simple, is that we wanted participants to try to use Stepic, because it's not a lot of them are doing contests. Not a lot of them are doing learn courses from Stepic and so on. So the qualification round, it's not just to limit the number of participants in the finals. It's also like a lot of it is for participants to learn how to use a platform, what is a competition, and so on. One thing that we didn't expect was that, well, we probably should have expected, but didn't thought about it, is that a lot of participants in our contest didn't took part in any other contests. So the whole idea of the programming contests were not that obvious for them. So that's basically why the qualification round was devised.
Yeah, the reason behind the qualification round was more educational. It's like what Alexey was talking about. We wanted to attract biologists to this context, not only computer scientists, and we wanted to get them used to how the test system works, and etc. Plus, to add some more of competition into their contest.
And actually, Stepic works just fine with 1,000 participants, so that wasn't the problem.
Mm-hmm. Yeah, so you mentioned Stepic, and for those who are not aware, Stepic is, well, first it's the platform on which the contest was held, and generally it's sort of like Russian Coursera, although it's not strictly in Russian, but has, I guess, roots in Russia. Is that a fair way to characterize it?
Yeah.
Yeah.
So what is your relationship with Stepic?
Well, one of the contest organizers is founder of Stepic. So we're pretty much in connection with the Stepic team. And yes, while we developed the first bioinformatics contest, there was a huge interest from the Stepic team to organized an international competition on their platform. So our interests has met. And also Bioinformatics Institute, apart from the contest, we develop several online courses, mostly in Russian, on programming, bioinformatics, biology. And we create them on Stepic for a wider audience. They're all open and free. And we've been creating them since 2014. But before the context, we have never used this platform for anything else than online courses. So it was new to us.
How good is the qualification round at filtering participants? Do you have some statistics? How many people took part in the qualification round, and then how many people progressed to the final round?
So it was... The statistics more or less this. So there are 3,000 enrolled people that basically said that I want to enroll to the competition on Stepic platform. However, only, I believe, not only, around 800 of them actually did take part in the contest. So they tried to submit something and got some points. And around 400 of them qualified to the finals.
But then what's interesting is that I looked at the final table from the final round. I don't know, does the final table include those who did not get into the final, or is just those who qualified for the final?
It's all people that qualified to the finals. So not all of them actually did submit anything.
Yeah, that's the thing. I think over half of participants didn't score a single point. And do you have any insight whether they tried to submit something but failed, or they just maybe forgot about the contest or didn't care?
I believe the latter, because there was pretty easy steps, like parts of the problems that was easy to solve. So I believe it's not hardness of the final. So basically, both qualification and finals are designed that they have simple problems. So maybe that they're not enough to qualify or to get to the top 20, but it's we try to make it interesting for all the people. So not only the top ones, but for all of them. So I believe it just they didn't want that much. Maybe they had some other stuff to do and so on.
Yeah, I wanted to add that maybe the timing was not perfect. We tried to check before announcing the date that we are not overlapping with anything related to the contest or programming or in biology. But there was an Olympics finishing on this day, I guess, like the final hockey match or something. And there were a couple of competitions. They still overlap with us. So maybe some people were more interested with other events.
Actually, so I was just talking to Katya about this. So Gennady Kurtkevich, I believe, Roman, you know this. It's you know this guy, and he was their champion and the last time, the first time, and also known in competitive programming. So he actually took part in 2 other contests during our bioinformatics final round.
Oh, wow, that's incredible. Yeah, I have my own story about Gennady Kurtkevich. I actually don't know him personally, and so the story was that last year he also took part, and I think he scored the first place. And I was showing this contest to a friend of mine who is into these programming competitions, because I'm not a big fan of programming competitions, but I showed these problems to a friend of mine, and he looked at the scoreboard and he's like, "Oh, Gennady Kurtkevich." And I didn't know this guy, but my friend told me that he's like a super genius. Is he also from St. Petersburg? Or I think he's Belarusian, but he studies in St. Petersburg or something?
Yeah, he was born in Gomel and he's Belarusian, but he studies in ITMO. So he won 2 times the World Programming Championship as a part with a team from ITMO and so on. So he finishes his master degree now.
Do you know if he's also into biology, or does he consider this just as a programming challenge?
Well, he doesn't do biology. That's for real, sure. But so basically his master thesis will be about actually one of the bioinformatics related problems about gene set enrichment analysis. Yeah, and basically actually I will. I'm going to be his supervisor on this. So,
very cool.
Yeah, but this is like the thing that we want to say that while he is from ITMO and I know him personally, we didn't help him to take part in the finals, so he's on his own.
Oh, of course, and yeah, I'm sure he doesn't need any help.
Yeah, actually, he could help us. But he decided to participate.
Yeah. So obviously you try to balance your problems, include some hard ones, include some easier ones. How good are you guys at predicting which problems will turn out to be hard and which will turn out to be easy? Didn't ever happen that you thought that some problem was an easy one and very few people solved it, or you thought that a problem was a very hard one, but a lot of people solved it?
Well, the hardness is mostly, predict it more or less good. like one thing that we didn't do properly here in this round is the fourth problem, where actually not the problem was not about hardness but about scoring scheme. So it was pretty easy to score something. And however, otherwise the hardness can be predicted rather well. The thing that we want to balance, aside from hardness, is that biology and informatics. So that's harder to predict. Basically, our first, the previous year, the most problems were algorithmic. So it was not really it was bioinformatics contest, but it also can be viewed as a programming contest in bioinformatics. However, this time we tried to do it to make it more bioinformatic-ish. I don't know. So the first problem was required at least Googling some biology or knowing some biology.
Yeah, I think that's fair. I also noticed that last year it was pretty much programming competition problems, but sort of with this legend, right? For some reason, these people who write problems for programming competitions, not just this one, but in general, something I noticed and something I don't quite like about these competitions is that they like to write really long stories, right? Wrapping this more or less simple to understand programming problems, but they like to wrap it in this very long legend. And it always takes me quite a bit of time to decipher what they actually want me to do. And so last year I got this impression that this was pretty much, a programming competition, but with the legends being about some biology. But this year, I think it's fair to say, you improved quite a bit on that. Yeah, the problems actually look like something a bioinformatician would solve. And I think there is a spectrum, right? Some of the problems were very artificial, I would say. But some of them, so for example, tandem repeats was pretty much a bioinformatics problem, exactly as a bioinformatician would solve it. And there are a lot of papers written about it.
So as you mentioned in the tandem repeats, so I want to thank David Eccles. So for the previous year, all the problems were designed by our team. However, this year we made an open call for problems. So and David responded, and this problem for tandem repeats is actually based on his ideas. So, and he is a bioinformatician. So, and also he was saying that our previous contest was just about programming. So, yeah.
Cool. So what about this year's problems? How many were made by you, and how many were contributed?
Yeah, so this year we didn't have a lot of problem responses from open call. So basically, the one we selected was only from David Eccles. However, we expanded our team who devises the problems. So basically, I maybe try to talk about the organizational part. So there are two sets of people. So one of them are designing problem ideas and maybe sometimes legends. So basically, what's the problem will be about and how it will be solved, probably. And there is another team, basically the guys that have big background in contests. So they know how to do tests. So they design tests, they design solvers, they design checkers, and so on.
Yeah, so this year the team included both biologists and computer scientists, all familiar with bioinformatics.
Yeah, but we hope for more responses for our call for problems next year.
Now, if someone submits a problem, isn't that sort of a conflict with their own participation? How does that work? Like if I submit a program that I already solved, or at least I have plenty of time to solve it.
Well, basically, if you submit a problem, you agree that you won't participate if this problem is in the finals. So this year we decided, probably the next few years also, we decided that, we had this, problem, conflict of interest for David. So we decided that the problem as his problem is in the qualification, well, he can solve it actually, so there was no problem for him to solve it.
Right.
Basically, we believed he will be qualified.
Yeah.
And actually, he was qualified the last time, so there was no problem with this. However, if this problem would be in the final round, so he wouldn't be. Well, either one is have problem in the finals, or he doesn't participate in final, or he participated in finals. Yeah.
So that's an additional sort of disincentive to submit problems, because if you happen to be lucky, you will be excluded.
Yeah. Well, in any way you can say that. Well, please get my problem back from the problem set. I want to participate. So, and at least it's like for qualification round, it's also not trivial to make a problem for qualification. So it's always a way to go just to make it in the qualification.
Yeah. So once you have, for example, once you come up with a problem, then you have to come up with a solution, right? And I guess for some problems, right, there could be different modes of coming up with a problem. Sometimes when you come up with a problem, just the fact that you came up with it means that you already know the solution. So for example, you can design a problem around a known algorithm that you have in mind, right? Other times you can design a problem, and even if you don't have a solution to that problem, but because you are making the tests, this is sort of this P versus NP dilemma, right? Because you're making the tests, you know the solution. So a good example was the problem with phylogenetic networks, right? Where you had to color the networks, but if you design the test case, you know the coloring, you know the true clustering, or at least a feasible clustering. But there is a third option, which is you have to solve the problem, and it may be a hard problem, right? Do you ever struggle to solve your own problems?
Well, we have a motto, is that it's participants. Well, the problems are solved by participants.
Right.
So usually a must is to have at least idea of a solution. So for example, for this cattle breeding problem, actually we don't know what is the best way to solve it, at least in this setting. However, we know that how it could be solved. And it can be seen that there are no 100% points for this problem. As basically we overestimated a bit how much it can be optimized. However, and this is not a ideal scenario. So usually, for example, for haplotype phasing, it's also not. It's a hard problem. However, there we had a lot of solutions. And we designed it in such a way with such tests that it can be possible to solve it. And basically that's, you can see that there are a lot of people, well, not a lot of, but top 10 people solved it 100%.
But for some problems, for example, the optimization problems, right? Unless you have the correct solution, you can't really score because you don't know whether the participant's solution is the optimal answer or not, right? So you have to solve it.
Well, again, for cattle breeding problem, the points were set, the scoring scheme was set because we knew upper bound more or less on the solution. So we didn't know how good you can get to it.
Yeah, I'm thinking more, for example, in the qualification round, there was this restriction size problem where you have to modify the smallest number of nucleotides. And so you have to know what the smallest number actually is.
Yeah, and for this problem, we just had a solution that we know that was optimal, and then just compared it with it. So basically, for all the problems, we try to have a best solution. And however, we don't actually, not for all the problems we really need this. So with some of the it's actually a good part of the contest is that for optimization problems, some of the participants can get better scores than you can get. And actually it's good. It's a good thing. Like, again, for some other contests, if there is optimization problem, like the one guy, I know, like Kaggle competitions in machine learning. So the one who fits the model, the tests best is a champion. So we also can have it here. So we don't know how to perfectly solve, clustering problem, like from scratch. We know the answer, but we don't know what is the best algorithm for this. But there are participants who can get a lot of different schemes for solving this. And actually, some of them can be much better than because there are a lot of participants and they are very smart. So there is a limited team of contests. So it's actually pretty interesting to see how good they can get. Yeah.
It's even harder for you to solve these problems for the reason that you are the sort of reference source. So, for example, when I'm solving these problems, I can come up with a solution. I don't have to test it thoroughly. I can just submit it. If it's right, I get 100 points. If it's not right, well, I get a fraction of points, and then I know I have a mistake. So I go back and figure out where I did wrong. But for you guys, you have to know the correct solution, especially, well, as we discussed, maybe not for the optimization problems where the scoring is how well you optimize. But for the exact problems where you have to submit the exact answer, well, you'd better be sure that your answer is the correct one.
Well, and for this, we have 2 world champions in programming. So like Ilyas Banyan and Artem Vasilyev are out there for us.
So do you just give them problems? Don't tell them the solution or anything, but like go ahead and solve this, and then you check that their answers coincide or something?
Well, we basically discuss the problem with them, like basically how they would solve it, how fast they can solve it, and then from that we can set constraints for the problems, like how big the numbers and so on. So it can be so that it can limit what the solutions are good and what are not. And basically then it's the first part. So we just think what is the best algorithm for this, and the best means that correct and the fastest one. Mm-hmm. Well, at least the fast one. And then also ideally is that for the same problem there are several solutions written by several people, so that we indeed can check that all of them coincides and all of them understand the problem in the same way, and there are no hidden problems and no so on. So it's basically the practice from normal programming competitions. So high-level competitions have several solutions, several checkers, and so on. So,
do you have any experience in organizing other like programming competitions, or is this your first project?
Organizationally, I was like volunteering. So in 2013, I was volunteering for ACM ICPC World Finals that were held in St. Petersburg, and I also helped during the semifinals here, and also took part myself as a contestant in such competitions. And also Vitaly Aksionov, who is the master of the problems of the Bioinformatics contest, he also organizes competitions, smaller competitions in programming. And there are also the people from the team like Ilya and so on. They do the programming contests, they do a lot of training and so on. So it's basically it was a good thing that Katya came to us for this.
Is organizing the contest just for fun for you guys, or do you set some sort of goals that you try to achieve with it? And how do you judge whether this or that contest was a success?
Yeah, for us, well, I would say for us in the beginning, it was just for fun. Like we came with this idea that it would be cool to organize a bioinformatics contest. And luckily we met the ICMA University team to actually develop the problems. The idea behind this was to test, is it really possible to do something like to organize a contest in bioinformatics, to solve problems in 24 hours and to develop problems that could be solved that are kind of close to the real life, because most of them are not really exact. And also wanted to promote bioinformatics through computer scientists and biologists, like to attract attention to that.
Yeah, I think this more promotes bioinformatics among computer scientists because they look at these problems and the problems look familiar. The problems look like something they could solve even without like a deep biological knowledge. But then it's cool that you could contribute to biology research solving these problems or problems like these. Were there any lessons from the last year that you incorporated in this year? So something you learned, something that didn't go well or went well? How does this year's contest compare to the last year's from your point of view? One thing we already mentioned was that you tried to make the problems less sort of artificial, less about informatics, a bit more about biology. What else did you learn from the last year?
One thing that was important to remove problems where you have to submit a code. So just limits the number of participants. So it's not all of people familiar with this. So again, like in this time, all of the problems you could download the tests, solve them with whatever tools you have or want to use, and then submit the answer. This was a major change from the last time.
Well, the last year we did everything for the first time. So we tested a lot of things, starting from like basic organization and explaining the rules, like delivering the information, how it will be organized. And we had to decide from scratch how we would organize that. And this year it went, I would say, smoother, because we already knew the process and we needed only minor changes. And we knew how to communicate to participants. So I guess we improved our communication, so there were more newsletters and easier process of signing up.
And one change was the time zone change.
Yeah.
So we decided to start at noon UTC because it's 3 p.m. in Moscow, so we can start the contest, then resolve problems that appear fast from the start, and then probably get some sleep. Not much, but some sleep in between.
What was it last year?
The last year was midnight UTC. It's like 3 a.m. Like it was shifted 12 hours. So it started at 3 a.m. So it was pretty hard.
Yeah, it makes sense.
Yeah, I guess for minor changes, I also just remember that we also changed the qualification scoreboard to look at the same way as the final scoreboard would like, so people won't be confused how and where to look for their scores, and it appeared to be more, to give a better idea how we would monitor the results.
What were the numbers last year and this year? Did more people participate this year?
No, it was more or less the same. It was also around 3,000 overall people who signed up for the competition, and around 400, maybe a bit less, that qualified for the finals.
Yeah, and I noticed that the majority of names were sort of Russian-looking, so there's probably a large number of people that you could reach that would be interested in participating, but I guess they just don't know about the contest because the main people you can let know about the competition are in Russia, right?
Well, the last time, so we had a statistics. It was about 700 people from Russia, 700 from US, and like 500 from India. So it actually wasn't that much Russian contest. And this year, if you look at the top table, well, there are not that much Russian names.
Yeah, I just opened our table from last year with the statistics on users' location, and the domination is by United States. It's like 685, and followed by Russia, 470. That's true that our communication channels are mostly like Russian oriented, and we are known like in St. Petersburg and Moscow. But we also send in information about the contest through the Rosalind, the bioinformatics tasks portal. So, and there are lots of people from various locations there, but maybe also Rosalind is primarily Russian-based project. So maybe there is a connection with it.
I didn't know, by the way, that Rosalind was had Russian roots. That's interesting.
To my best knowledge, it's in the cooperation of the St. Petersburg Academic University, where Nikolai Vyahhi was working at the moment, and University in San Diego.
Yeah, and for those who don't know, that's rosalind.info, I believe. So you should check it out. There are these problems. Would you say those problems are similar to the problems from the contest, just maybe a bit easier, or is it a different style?
Well, they probably are similar to the first contest. So usually they're more like algorithmic bioinformatics. That's we tried to avoid this time. So a lot of problems this year didn't have, like in the finals, basically only one problem has an exact algorithm this final. And at Rosalind, there are problems for specific algorithm, for alignment, for tandem, small tandem repeats, and so like motive search. So while a lot of people come from Rosalind to the contest, it's not the other way, probably.
So let's talk about the problems. Shall we start with the qualification round? The qualification round lasted a week, and there were 3 problems. So the first problem of the qualification round was synthesis of ATP. And the legend was that you work in a biotech laboratory which specializes in production of ATP from glucose and oxygen using yeast S. perfectus. The species was developed so that it can very efficiently produce ATP using 2 pathways, fermentation and aerobic respiration. And so the laboratory has a budget, and you have the price of glucose and price of oxygen, and you have to figure out how much of each you should buy to maximize your production of ATP. And in the problem, which was a part of the problem, you're not given any reactions, so you have to figure it by yourself, but you are given a few examples. So a few inputs, a few outputs. Now, if you know linear programming and mixed integer linear programming, what they are, and just basically the terms, and you can Google them and you can find the library, that solves, I guess, the computational part of the problem. Then you just need to figure out the reactions. So what was your reasoning? Was that primarily directed at people who know about these algorithms, or at least their names, or was the idea that you have to figure out how to solve this without any special knowledge?
Well, we designed this problem to be like an easy problem for like all people. It's actually was our first try to get to like a bioinformatics problem. So as you said, there was no explanation what is fermentation, what is respiration, and so on. So for people who know this, like from biology background, well, in general, they maybe don't know the linear programming framework. However, here we had it like with 2 constraints, so it should be pretty simple to like draw the lines and so on and understand how to solve it. And we did a like harder version for the second part. So the main idea was to get like a computationally simple problem, but to have a like more biological, related knowledge to be required, or like at least Googling to be required to solve it.
That was what I did. I just Googled these reactions, and I arrived at the Wikipedia page or pages for aerobic respiration and fermentation. But the catch was that those reactions didn't quite work, right? And there were quite a few people complaining in the comments that the reaction is a bit off. So specifically, I believe the oxidation reaction, I think the Wikipedia page says that you have glucose and 6O2, and you get 36 molecules of ATP. But in your tests, it appears that the result was 38 molecules of ATP. And someone in the comments said that you probably sort of forgot to subtract 2 molecules of ATP that participate in the reaction itself. So was that just a mistake, or was there some reasoning behind 38?
So the textbook actually, so here we didn't try to make something sophisticated. So and this text, a lot of textbook say that the oxidative phosphorylation make 38 ATP per glucose and 6 molecules of O2. So and actually, so yeah, however, the same Wikipedia page says that actually the number is smaller, and for some organisms it's 36, for some of them it's 38, and so on. So it was quite hard choice to make, like which one should we put. So we put 38 and said that it's S. perfectus, basically the ideal bacteria, like yeast. And I believe after all of these complaints, like before all of these complaints and after all of these complaints, I was reading a lot about yeast metabolism. So I believe 38 is reachable with malate aspartate shuttle, which can work in yeast. But for this, again, as we made the problem, as I said, that it was not trivial to. While the number 38 appears in Google pretty easily, or 36, we just said that, okay, so the people, what they really need to Google know is that it's 1 glucose and 6 oxygen. And how much ATP is produced can be found from the tests, from the input samples. That we also assume that it's that it's common, scientific knowledge that fermentation is 1 glucose and 2 ATPs. However, the comment that fermentation can be get 4 ATPs in some weird circumstances. So there we didn't think about this. However, also the number 2 can be found from the second sample, from the samples for the harder version of the problem.
Yeah. That's how I basically. Yeah, that's how I solved it. So I couldn't Google the correct reaction, so I tried the standard one. It didn't work, and then I just wrote another program that enumerated all the reasonable coefficients in these reactions and compared the results to the official results, and then it just came out as 38.
Well, basically, after you know that it's 1 glucose, 6 oxygens, or 1 glucose per 2 ATPs, it's pretty simple to find that it's actually in the examples you need to buy a lot of oxygen and to get the respiration going on, and then just don't have money for glycol for fermentation. So there, number 38 you can get it pretty easily, or by enumeration or whatever.
Yeah, that's assuming you know which number is wrong, but usually you just submit and you see that your result is wrong, and you have no idea which one of the 6 or so, or maybe four coefficients is wrong. So I decided just to enumerate all of them. Okay, so problem 2 in the qualification round was restriction sites. And there were 3 versions of this problem. So the first 2, the first version was a special case of the second one, so I guess there's not much point in discussing the first one. But in the second one, you're given a primary RNA sequence, and you are given a set of intervals. And you have to change the smallest number of nucleotides in this RNA sequence such that each interval contains at least one changed nucleotide, but the translated amino acid sequence remains the same. And so the first version was a special case of the second version, where it was guaranteed that the intervals are non-overlapping, in the second version, they could be overlapping, and they were. And then the third version was that instead of just the intervals, you had a set of strings that shouldn't occur in the modified RNA. So you don't know where basically they can occur anywhere. So version 2, do you want to describe your vision of the solution, or I could describe mine?
Actually, just like a disclaimer. So I, not for all of these problems, I know exact solutions, exact algorithms, because it's hard to know everything. And so for this problem, I believe the simplest, the first and second version are pretty simple, and they are, it's kind of messy to implement, but it's pretty straightforward for each interval, or like 2 consecutive intervals, and you can check a few cases, maybe not a few, about whether you can change something or not, like using one single nucleotide change, or like two nucleotide change, and so on. And the hardest version is like dynamic programming, basically. Again, with some cases, but more or less the same, I believe, from pretty similar to the easier ones.
Interesting. I didn't think about dynamic programming, to be honest. But so the way I solved it is first I did some simplifications. So the very first step, very useful step, is just to remove redundant intervals. So if you have one interval nested in the other interval, then you know that if you satisfy the smaller interval, you'll automatically satisfy the bigger interval. So you can just get rid of them. And surprisingly, there were quite a few of these large intervals that had smaller ones inside them. So you could just remove them. And the reason this matters is because the second step I did was to split all the remaining intervals into connected components. So I could look separately at sort of these islands of intervals that do not intersect. Because my algorithm is not linear, it's worse than linear, and so you benefit from splitting this into groups, and removing these large intervals helps splitting into smaller groups. And then some of these intervals turn out to be singletons, so they are the only ones in their group, so they don't overlap with any other intervals. Also, it's worth noting that when you see whether 2 intervals overlap, this has to be aware of the reading frame and boundaries, because they might not overlap as intervals, but they may sort of touch the same codon, the same 3 nucleotides in the reading frame. So you have to take that into account. But yeah, so handling singleton intervals is very simple. And then in small groups that remain, I basically did branch and bound, which is you enumerate all the possibilities, but you enumerate them in a smart way. So because the full tree of options is huge. And so you try to prune that tree. So, for example, once you found a solution, right, every time your current score sort of exceeds, so your number of nucleotides exceeds that you already found, there's no point in exploring that branch because it's already worse. And then I did a simple feasibility check. So to prune the variants that are obviously not viable. Basically, I did a stupid search, but tried to prune the bad branches in a smart way, and that worked reasonably well.
So you really like branch and bound algorithms, I see.
Yeah. Well, I was correct. It's pretty much, I think, the first time in my life that I actually implemented one. So while I was thinking about this problem, and I had this huge tree that was very inefficient. So when I tried, my algorithm didn't finish in any reasonable time. So I was thinking how to optimize this. And then I thought, well, I should not explore the branches that are bad. And then somewhere in my sort of subconscious, I remember this term, branch and bound. And it just seemed relevant, you know, because probably I read or heard about it somewhere. the basic idea is very intuitive, and sort of once I figured that out, I just had to apply it to the specific problem. But it turned out very, very efficient. And then in version 3, the difference is that you don't have the intervals, but instead you have the sequences that you need to avoid. And so the naive way would be to use the previous solution, right, to find the intervals in which these sequences occur, these forbidden sequences, then apply the previous version to filter out, to mutate those intervals. But then, of course, some of the sequences may reappear because of your mutation, so you have to repeat and repeat. I'm not even sure whether this process necessarily converges, but that's sort of one way to approach this.
Well, I believe we have a exact solution. I believe it uses dynamic programming, probably like something like suffix trees or something like this, to check whether you will have something to appear or not. So I just say here that we'll publish the problem analysis with like solutions, not only with answers, but how to solve it. I believe maybe by the end of the week or maybe the next week. So all the solutions that we have, exact ones, will be published, so that you can look how we try to solve it, to approach it. And I believe for this problem we had like an exact algorithm that runs in polynomial time, and that's why these constraints are coming from our jury solution. So that they're not, because in the like branching bound or stuff or like approaches like this, so you don't have a exact upper bound on how much time it will be required for the problem.
You have to be lucky.
Yeah. So probably we didn't do the test properly for this. So actually that's why. So usually, if like contest team, like in general, if the contest team has like a lot of time and people, so that's why the same problem is approached by several people, and they maybe even like the same person the same team can do several solutions, so they can be graded differently. So for example, these constraints will differentiate between that simple solution and the harder one, but faster.
So basically, if you knew that I would do a branch and bound solution, you would come up with a test that would penalize me and sort of make that infeasible, right? And make me come up with a dynamic programming
so it's actually relevant. So here in the lab, like the adjacent lab, we have research that does on evolutionary computations, like evolution algorithms and so on, like optimization. And one of the application is how to make the hard test for NP hard problems. So there is one site like called Timus in Russia, like made by Ural Federal University. They had it's like Rosalind. It has a lot of problems, but in algorithms, and several of the problems are actually NP complete. So the constraints are not that big, but kind of biggish for NP complete problem. And so several of these problems had some solutions that passed the time limits for the test that they had. However, and so the guys that were doing it here, like adjacent to us, they tried to, for particular solution, to try to find a like test case that will fail this problem, so that it will work a larger times than the time limit. So yeah, if we knew your solution, we'll be trying to make a proper test for this.
Yeah, and for version 3, which is about mutating so that no substrings appear in the RNA string, I also used branch and bound. So every time you use dynamic programming, I use branch and bound. And the way I did that was so as you mentioned, you can use some kind of tree. So I used Achocorsic algorithm to so you can build this sort of tree out of your forbidden strings, and you can traverse the string, and you can determine in linear time whether the substrings appear anywhere along your way. And so you can produce the mutations, right? But of course, then you have to minimize the number of mutations. So again, you have the search problem. And so the way I did branch and bound here, I had to come up with a good bound, because otherwise, if you're in the beginning of the string, it's very easy to come up with some solution, but then you have to minimize it. And so let's say this some solution is 100 nucleotides have to be mutated, and then you're looking for a smaller number. But the problem is that when you're in the beginning of the string, you have this huge budget of 100 nucleotides. And so you can be very wasteful with them. But then once you approach the end of the string, you realize that that was wasteful. But you already spent a lot of time exploring those branches. And so the way I resolved that was I split the string in two, right? And again, because this is a worse than linear algorithm, it runs much faster on, let's say, half of the string. And so I run the algorithm on the second half of the string. And of course, that recursively runs it on the last quarter of the string and last eighth of the string and so on. And so this allows me to get a more realistic bound, because I know that this is a sort of minimum bound on how many nucleotides I have to mutate for the second half of the string. And then I can incorporate that into my bound calculation. So that worked reasonably well. I also had, speaking of NP-complete, the way I approached the first and second versions of this problem, where you have just intervals. So first I formulated this as an integer programming problem, right, in the spirit of the first problem, where it was mixed integer linear programming. So I figured, well, I could use integer programming here too. So the way it works is you can enumerate all the codons and all the mutations in those codons. And then you have these constraints that each interval has to have at least one mutation. So that's just a linear constraint, right? The sum of so basically you assign 0 or 1 to every codon. And you say that at least one of them has to be mutated in each interval. And then you also come up with a constraint that says that each codon has to be mutated in at most one way, or exactly one codon sequence has to be assigned to that triplet. So I tried that, and it actually worked okay on the smaller examples. But the fun thing was that I submitted my solution, and most of the responses were correct, but like a tiny minority were wrong. And that was odd. And then I reran my algorithm, and some of those actually changed. And I submitted the second time, and I got a higher score. So they were sort of changed for the better ones. and the algorithm was completely deterministic, and I used this mixed integer linear programming library, CBC, and its bindings for Haskell. So I solved all my problems in Haskell. And so I used this library that brought this, and CBC is written in C++. So I was thinking, and CBC is like this very cool library, which is, I think it's maintained at IBM by some cutting edge researchers. So I thought, well, there's clearly a bug somewhere in the library, because, well, my program is completely deterministic, right? And so the problem could be in C++ library, but then this library is widely used and widely tested, and that seems unlikely. Or the problem is in the Haskell library that wraps the C++ library. But the Haskell library is very simple. All it does is it calls C++ functions. So that was very mysterious. And after I already solved this with branch and bound, but I looked further into it, and it turned out that the Haskell bindings had a bug where it was basically a memory management bug, because you have to explicitly call all the constructors and destructors. And but in Haskell, I guess they use this automatic release. And so because they didn't declare the dependency on the structure was sometimes occasionally released prematurely, and that resulted in memory corruption, and that resulted in a somewhat wrong solution. So that was fun.
So was it fixed at the end?
Yeah, the guy fixed it very, very quickly, and I think so it was like within a day, and of course the qualification route lasted a week. So I had plenty of time to use the fixed version to solve the first problem. I no longer use it for the second problem.
So that's like a good result of bioinformatics contest fixes.
Oh yeah, yeah, that was excellent.
Fixed.
I guess the main result for me is, well, first I found and reported this bug, which got fixed. But also what I liked about this is it makes me tackle some problems that I wouldn't find in real life. And not just, read some papers. So, for example, tandem repeats, which we will discuss next. I didn't solve that problem, but I looked into it, and that made me find and read some papers and try to think how I might implement that. And this is a totally different level from just, casually reading a paper about an algorithm and, it sort of makes sense, right? I probably could figure that out. But actually sit down and do figure it out, or figure out how to use a branch and bound algorithm, that's actually very, very useful. And so I really got a lot out of this competition. So thank you.
You're welcome.
Okay, so problem 3, tandem repeats, right? And it's basically what you would imagine tandem repeats are, right? So they are this repeated sequences that occur next to each other. So you have a long string and then the exact same long string right next to it, and these all embedded in some very long genome or whatever, and you have to find them. And in this case, they were not exact, or I guess were they exact in one of the versions, maybe?
Yeah, the simplest one was the exact one.
But in general, they are not. They're not exact, and they are measured by their Levenstein distance, which is basically the added distance, right? How many nucleotides you have to either replace or insert or delete to make them the same string. And there was a constraint, because obviously you can mutate any string to any other, but there was a constraint that the added distance had to be less than 10% of the length.
Of the
right, I think it was the
maximal
min length. Max length?
Yeah,
okay.
One of this.
This problem, you said, was proposed by David Eccles. Did he also submit a solution to this, or did you have to solve it yourself?
So actually, this problem, I believe we didn't have a proper algorithm to solve it. So we tried to. Our problem was to make tests here. So and we tried to make it so it can be solved in different ways. So with the exact problem is pretty easy. So there is just it's a standard problem from discrete mathematics. So normal contests where you can, or maybe actually not that standard. So, but the main idea was that people can approach this problem from different perspectives. So one of them is just they can use blasts or something, and they can use NukeMare. They can write some programs, algorithms for themselves, and so on. So here, I believe we didn't have an ideal solution that we tried to achieve. So it was nice to see. I believe there are several people solved it 100%. So it's pretty nice to see. So we are not, actually, I believe we're not sure that it's how to find all of these repeats. Of course, as we generated tests, we knew what is the possible answer, but how to get it, I believe not for all those tests.
Yeah, and then of course there's a tiny possibility that there's an even longer answer that you don't know about.
Yeah, and we didn't restrict it this way. The longer answer is also okay, so it's get 100%.
Yeah,
so
of
it's
course.
not that it gives you something. So that's like one of the limitation, basically, like for this problem and for other optimization problems. So we had, I don't know, probably maybe we'll switch from this scheme, but there's another scheme like aside from just scoring, like assigning some scores for some values of basically your objective function, is to compare participants with each other. So it's more complicated to, well, it's slightly complicated to implement, but not that hard when we have a separate monitor. But it's probably harder for participants because the scores kind of depends on the time. But it's, I know, probably for some of the problem next time, maybe you'll do something like this, so that basically your score is how good are you compared to the best to the best solution among the participants.
Yeah, from the participant point of view, that's like very frustrating and depressing. If basically your score can only go down, right? Once you submitted it.
Well, it can't go up until. But yeah, So we discuss it among ourselves, like what would be easier and simple to understand and more interesting to have. So for this problem, it's actually pretty easy. For this problem, it's kind of okayish to use current scheme. I would say that even the scheme is pretty good for this problem because you can't get much better than the answers that we have. And also the answer that we have is pretty achievable. So this is compared to some other problems where the landscape is tough to understand.
Do you know if any of the standard tools for tandem repeats could do well in this problem? Like if someone just downloaded and compiled some tool from GitHub or something?
Well, I believe one of the things that worked for some of the people were using NukeMare. NukeMare is a linear of sequences. So and it can plot a dot plot, I believe it's called. So just like describes how fragments of sequence is aligned to other fragment of sequence. So and there, if you have a big line, well, there you can see if you have a repeat. I would say this. So I believe it worked for some of the people. So we didn't try on this, but we did expect for people to be able to solve it this way.
But in general, when you design a problem, does that play a role whether there are any existing tools to solve this problem? Do you try to avoid it or maybe try to welcome it?
Well, it depends on the problem. So usually we try to do the problem in such a way that it can be approachable with some of the available tools, at least, because so that's mostly coming from the idea that you want a lot of participants with different backgrounds that maybe they don't know how to. Actually, they probably don't even know how to write programs. If you just download the input, it's just faster and use some program that you know how to use. And it's where bioinformatics comes from. So if you know how to use it, well, you can use it. And so we are try to be aware of the tools, and at least for the simpler tests, we are actually trying to make them be solvable by existing tools.
Okay, so that was the qualification round, and now the final round. So the final round, first problem was recombination of plasmids. That's another one of these very vague problems where you sort of leave it up to the participants to figure things out. So like the first problem of the qualification round. So in this case, the text said, DNA recombination involves the exchange of genetic material either between multiple chromosomes or between different regions of the same chromosome. And this process is generally mediated by homology, that is, homologous regions of chromosomes line up in preparation for exchange, and some degree of sequence identity is required. Recombination is very important as it takes part in DNA repairment and gametogenesis. Recombination is widely applied in genetic engineering. In this problem, we ask you to use one of the types of recombination to assemble a plasmid. A plasmid is a DNA molecule commonly used in genetic engineering to deliver some genes from one organism to another. The classical way of plasmid assembly is restriction ligation method that requires cutting plasmids with special enzymes called restrictases and then putting the fragments back together in the desired order with enzymes called ligases. However, in this problem, plasmid assembly requires recombination between different plasmids. One of them provides the scaffold and others, one or more, contain DNA sequences that need to be arranged in a certain order, one after another, during a process called site-specific recombination. And so the solution to this problem is the plasmid sequence resulting from site-specific recombination between p-dest sequence and p-enter sequences. It has to contain sequences from each p-enter in a specific order inside p-dest. So that's a very long story that pretty much tells you nothing, except I guess the keywords site-specific recombination. I have to say that this problem was like super frustrating for me. Like it seems that pretty much, well, not everyone, but like a lot of people solved it, and I just couldn't. Right? And I was sort of near the top of the table, and like all the people around me in the table solved this problem, and I couldn't. So tell me what it is.
Yeah, so it's again, as people nicely commented in the comments, that it's a reincarnation of the ATP problem. The idea again was that we don't have like formal statement that says what you need to do. So, and from this you should Google stuff like recombination, p-dest. Actually, this is a nice keyword.
Oh, this was a real thing, because I thought that was just a sort of variable name or something that you put in there.
No, actually no. So it's the names like this are used in the plasmid recombination field, I would say. So, like actually for this problem, I didn't know the solution, so I tried to solve it myself. So yeah, I was Googling some site-specific recombination, something else, p-dest, p-enter, and so on. So I was trying to find, because you are given the sequence, so like two sequences you need to recombine. So I was trying to align them on each other or between them. So I'm probably looking for some repeats, because the recombinations they should have some repeats to recombine. Well, not repeat, the same sequences to recombine. So there was no big overlaps. So like 7 nucleotides something. So I was Googling again, then found some ATT sequences that I was able to find in the sequences that are in the input, and also like found some images saying how these one things go to, like how they recombine. So at some point, word like phrase gateway recombination appeared. So I googled this. So there was some nice pictures how it works, and actually so from this, you can Google some sequences. It's called like ATT sites. Basically in p-dest there was, I believe, ATT L1 site and ATT R1 site from like left and right. And in another sequence, you have ATT L2 and ATT R2. So also like left and right. And the first one is recombines with the first one, like ATT L1 recombines with what? Yeah, ATT L1 recombines with ATT L2, or it's hard problem. So like after looking for all the sequences, and looking at the figures that like there are some materials on, I believe, Thermo Fisher website and some other websites. So from this you can understand like how, in what order, that ATT sequences are changed to ATB1 sequence and ATB2, and the one fragment is like coming to another DEST sequence and so on. So like after fondling with this for a while, I was able to get the simple output, like the proper answer for the first problem. It was harder to get for the hard version, because there you have actually 4 plasmids. Because so it works like you have 2 plasmids, like in the easy part, and they have some similar sequences that are like repeats between both plasmids, and so they at these places the plasmid recombines, like the 2 of them get together and recombine the sequence. However, in the hard version, there are like four plasmids, and not just they recombine like one with one, and then so the result with the second one, and so on. But they actually recombine all at the same time. So again, you can Google like 3 fragments gateway recombination or something like this. So they are also nice pictures how it works. I wasn't able to make it myself. When we followed like how participants solved it, we were not sure that it will be like for like how solvable it would be. So when we saw that, well, people do solve it, then okay, that's great.
Yeah, that's always a relief.
Yeah, that we didn't make it super hard and unsolvable. Yeah. So, and actually, in this problem, there are several tools to maybe not several, like maybe a lot of tools to do this. So, however, not all of them. So I was trying to use one of them. It didn't work for me because some like these sites were weirdly found. It was found as ATT sites, but something happened. However, I believe we know that several tools, at least one of them at least, can be used to do this. So, and it's like one of the standard instrument in like biotechnology. So people do this. It was very nice that so now, while we don't have analysis published yet, so there is some discussion in the comments. So there, Kirill Trukhanov and I believe Roberto Rosati, who solved this problem, they have published in comments how did they solve it. So there a nice discussion. So at first I thought that, well, these guys probably just knew how to, like, are familiar with this and knew how to do this. But actually no, they Googled a lot of stuff and found that how it works. So inside the team we discussed like during the contest that probably we did like a harsh scoring scheme. So that's either like 0 or 700. So probably it wasn't the best choice, but like actually it was solvable even without the proper knowledge how to do this. So people actually did Google it and found how it works. So it was pretty great.
Yeah. So my mistake probably was not enough Googling. So I thought I'd just Google one query and find the solution. But apparently, you had to dig really, really deep into this to figure it out.
Well, actually, I believe so. When I did, something came up in like 10 minutes after of Googling. So basically, the idea is that once you are, so basically for recommendation site, or for site-specific recommendation, there are not too many ways to do it. And actually one of the links, it says like recommendation site sequences, where it contains some sequences that you can look up in your sequence, in the input sequence. And from this, it's kind of not very simple, but pretty straightforward. That you know that, okay, it's about ATT recombination, so yeah, we can work with this.
Yeah, for some reason, the stuff I found on Google was like very, very high level, not specific. There were no like specific sequences or anything. It's just they align and recombine, and that's how it's done. Anyway, so how did you come up with the examples? You said you didn't know the solution. So was that just another person who knew the solution and came up with the examples, or were these, the test cases, were they like actual real data that you somehow knew?
They were generated.
So generated by like some tool, or one of you guys generated it?
It was generated by tools and one of the guys. So it's basically the problem was designed by Alexander Tkachenko, like who does like biotechnology in his spare time. I mean, not spare. So he like devised these plasmids and sequences and so on. So basically that's how you do in biotechnology. So you have one plasmid, you want to get some sequences to another plasmid, and these ATC sequences are like in place. It's probably even based on some real plasmids. Actually, in one of the tools that are available, there are like it's a store, like app store of plasmids that actually calls like pdest and so on. Oh, and penter, like pdest 8 or something. So that's the pdest and penter was important keywords.
That's cool. Okay, so problem 2 in the final round was species recovering. And there you were basically given a directed acyclic graph, which is sort of a graph version of a phylogenetic tree. So you could call it like a phylogenetic graph. And in the leaves of this DAG, of this graph, you had, so some of them were marked with some colors that are like traits. And then you have to figure out how to extend that coloring up the tree so that there is no homoplasy. There are no traits that sort of independently arise in multiple branches, right? Is that a fair way to put it?
Mm-hmm.
Yeah, so did you have a good solution for this problem?
So there is 3 parts of the problems ordered by hardness. So the first one is, I believe, pretty straightforward. So it's actually you just try to color it and it either works or not. The second part is harder. There is also like exact solution, but not the trivial. And I believe for the third one, we don't have exact solution. The problem maybe is even NP complete. So it's actually hard to get this. Well, it sounds NP complete. So for this, we didn't have, I believe we didn't have an exact solution. This problem appeared because actually this was designed from the algorithm. So there was like a nice algorithm to solve it for the second version. So then we simplified it for the first version and made it harder for the third one.
Yeah. So what was the name of the algorithm?
I'm not sure it's named yet.
So did you just like invent it or something?
It comes from Max Alexeyev lab from in Washington University. They do some computational biology on these networks and phylogenetic networks, trees, and so on. So I'm not sure they published it, but they knew it at least, and maybe devised it. So one of the guys from this lab suggested this problem, and here it goes. I don't believe it has a name.
Oh, we should. What's the name of the guy? Alexeyev? We should call it Alexeyev algorithm.
Yeah, probably there was 2 Alexeyevs. So Nikita Alexeyev is one. So Nikita Alexeyev is one of the person on the team. So he developed this problem, problems with haplotypes, and linear programming problems for ATP from the qualification. So he was a postdoc in Max Alexeyev lab in Washington. Yeah, and they have different surnames actually. They sound the same, but they are written different. So probably it's one of the Alexeyev's algorithms.
So the third problem is haplotype phasing, and there you are given a bunch of reads. The reads have in them like actual variants and also noise. Mutations, insertions, deletions, and you are given the locations of the known variants that you are interested in. Actually, the number of variants was relatively small, so it was like 10 in some problems, up to 30 in the others. And the task was to figure out the haplotypes, and you are given the number of haplotypes. So basically, when you produce a read, you choose randomly, but according to some probabilities. You choose one of the haplotypes, and then you apply mutations and deletions and insertions and generate a read from that haplotype. And so you had to recover all the haplotypes. And the number of haplotypes was also relatively small. So it was, I think, 4 to 6.
Or 5, something like this.
Yeah. What do you think of that problem?
So it's again a problem that tried to be designed in a way to come from bioinformatics. So the phasing problem is pretty important, and a lot of people do this, and also becomes doable with long reads like bioreads or like nanopore reads and so on. So here we had a few different versions of this in the beginning. So whether their sites are known, whether they're not, whether they have to be aligned and found and so on. So we decided that we'll give the participants the places, like the sites that have different variants. So that it's simpler on the first part, you don't need to find them first. And then the problem is actually about trying to recover the haplotypes. And here, again, there is no exact solution for this problem. It's probably pretty hard. And the tests were designed for different sets of solutions. So, for example, the first test, actually, you can just enumerate all the. So it was pretty small. I believe there were 2 haplotypes and 10 places. You can enumerate all possible haplotypes and then get the 2 of them that are most likely to be from the data. And the next, they're getting harder and harder. I'm not sure how we solved the seventh test. So we had a solution. So for this problem, we had a solution that achieved 100% for all the test cases. So I don't know it, sadly. I believe we'll publish it in their analysis. And I believe you also have some nice solution for all of this.
Yeah, so...
or was it also branch and bound?
No. So it was even worse. It was pretty much manual labor. So I saw that a lot of people solved it, so it couldn't be hard. And also the test cases were pretty small. And at first I had no idea how to approach it. So the first step is... Did I do assembly? I guess, yeah, you have to assemble them and align them. Oh, no, you don't have to, because you were actually given the reference genome, so you don't have to do assembly.
Yeah. Yes, so that's one of the choices that we made. That, it's not a problem about genome assembly, so we'll have a reference
right
in the statement.
Yeah, so I just used Minimap to align the reads to the genome. First, I tried Bowtype. Bowtype was pretty slow. Minimap 2 was much faster. So I used Minimap to align. And then I couldn't actually figure out how to use any of the sort of variant calling or whatever tools to extract the counts. So I tried some tools, MPyLab, but it didn't quite work for me. Couldn't figure out how to use it properly. So I did all that algorithmically, just in my Haskell program. And so what I did was in my Haskell program, I just generated k-mers. So if you ignore all the rest, once the reads are aligned, you don't care about anything except those positions. So you can just sort of throw that away. And you have just this 10 or so positions. So you can treat this as a string of 10 nucleotides or 30 nucleotides. And then it's similar to how genome assembly works. So they use De Bruijn graph. So they consider sort of consecutive k-mers. And so in this case, I also printed consecutive k-mers. But then I was misled by... so in the problem, you said that there are certain frequencies for each haplotype. And so I try to somehow use that information. So I try to come up with some sort of probabilistic model that would tell me which sort of combinations are more likely. And I just couldn't figure out how to do that. So And this was like late in the night, so I took advantage of all the 24 hours. I got no sleep that day. And so late into the night, I would print these K-mers and assemble them manually, just on with pen and paper. And it worked fine with smaller problems, but with larger problems, so I was able to assemble something, but it didn't get me the full score. And then it was already the morning, so it was like 9 in the morning where it occurred to me that it's completely stupid because, well, I tried to assemble them all simultaneously. But what you can do is once you assemble the most likely one, you just throw away all the reads that are likely to come from that haplotype. And then you repeat the procedure. So then you assemble the sort of the second most likely one. And that makes it much easier. And I got all of them like exactly correct. But first I spent like probably the whole night like assembling them on paper.
So that's like reminds me. So Bioinformatics Institute it held Bioinformatics summer school in Russia. So and since I believe like since a few years ago, they started to make a genome assembly problem where people have to to assemble genome on like pieces of paper. So the reads are pieces of paper, and then they have to manually align them and so on. So I know.
Yeah, I also felt like one of those very early bioinformaticians or biologists who were aligning. I think I read about this somewhere that they were aligning reads like literally by hand before there were like powerful computers or something, or when they didn't have access to computers. They would just figure out on paper how to align them. So I felt a bit like that. Let's quickly finish the last two problems. Okay, so cluster the reads. That's basically a standard problem. So you have 16S rRNA sequences, and you have to assemble OTUs. That's a standard problem, except you made it harder because, like, usually those sequences are sort of all aligned, all have the same length, because when people do amplicon sequencing, so they have primers for specific parts of the 16S region, but you made it nanopore reads, which are not aligned. So I tried a couple of tools, but they didn't work. I tried USearch, the famous tool, but it really, I think, works on the fixed sequences, right? So do you know if any of the standard tools work on this?
I'm not sure. So like when we design tests, so one of the things that we tried to do is the length part is important. So the 16S are of the different lengths. So then the reads are of different lengths. So actually here we have some problem with scoring scheme. So the simplest solution that achieved like non-zero score was to get a separate read to separate cluster. So we didn't design this, so it was our mistake. However, like the second for test number 1 and a few other tests, so there you can just cluster the reads by the lengths. So there are like 3 in the first, I believe, test. There are like 3 actually like genes, different genes, and they are of different lengths, and they have different lengths of nanopore reads. Actually, it's not. It doesn't require actually any tool to do this. So there you can find lengths of the reads I believe pretty easily, and then cluster them together also pretty easily. Also, like for this problem, we don't have an exact solution like that achieved 100% score.
Mm-hmm.
So when we thought about this, we thought about solutions like calculating some kind of pairwise distance, or like maybe not pairwise, but to make a distance between reads. So the K-mer distance or something like this,
mm-hmm.
and then to use clustering, like available clustering algorithms, available implementation, or maybe not implemented by hand, like K-means. You can probably use it. Well, there are a lot of tools for clustering. This was probably maybe not for just standard tools for genomic field, but for standard tools in like machine learning maybe field.
Yeah. Well, so I used it with genomics tools. So what I did was first use an assembly program. So assemble these reads. I didn't realize. So are you saying every read covered the whole gene?
Not exactly, but it's close to this. Yeah.
So I used what did I use? I think MegaHit. I used just to assemble them and to get some context, and then I would use Blast to align the reads to the context, and so the reads that align to the same context I put into the same cluster.
Mm-hmm.
I think it worked reasonably well.
Is there like nanopore metagenomic something assembler? Probably you can run like Metaspades.
I actually, I have something to complain about because I tried to use Metaspades and it just crashed on me.
Oh,
that was very weird. Probably like I downloaded the precompiled binaries of Spades, and Spades just it just crashed when I tried to run it.
Well, you should report a bug like another one.
All right, and so the final problem is cattle breeding. Well, that was an insane problem. So at first it sounds like a very simple one because basically what you do, so you're given some cows and some bulls and some data about them, and you tell the server how to breed them, and the server tells you the result of breeding, and so your goal is to achieve the maximum milk output. And so first I thought, well, people used to do this. This is not hard, right? You just take the best performing cows and bulls and you cross them. Like what's hard about it? But then I tried to submit something and I realized that the catch was that you didn't report, like when you cross, so you say, for example, cross this 10 bulls and 10 cows, right? 10 pairs. And the server didn't tell you which offspring came from which pair. Right, that's probably was part of the problem.
It was line by line, actually.
Oh, was it? Well, I didn't realize that. Oh, gosh. Yeah, so how would you approach that problem? What was the hard thing about it, easy thing about it?
So there were two interesting things about it. So one of them is that, so that's coming basically from cattle breeding real problem. So you don't really know how to select bulls because they don't produce milk, so you can't select one that produces milk. So that's so for this we added like separate traits that probably can correlate with milk production. I'm not sure it worked for anyone, but they were not randomly. So actually they should correlate with milk production and so on.
Mm-hmm.
And the second hard part is that there were several like little recessive little like SNPs in the genome. So if you like inbreeding, basically if you select co-relatives, so at some point you can get like basically zero lifespan. That means that they are dead. But otherwise, I thought about this problem in general, like how to select, how to breed like cows and bulls, like what you should do with it, even like if you have a genome and if you don't. So it was kind of, I don't know how to solve it properly.
Yeah.
And we don't know.
Is that a business idea of yours?
Not, but I kind of know. I know guys who try to do cattle breeding, but
really?
with genome arrays, basically, to know the genotype. So, but anyway, like actually, one of the things that that we said here is that you can't cross more than 20 pairs in one submission. So it was also like not random, because in actual circumstances when you breed, you can't breed like anyone, like all the cows versus all the cows. So you have to select something like normal and then wait again for like a few years to know like how it works out and so on. So yeah, like for this, I don't know the solution. So, we encourage participants to say what are the algorithms that they use. But anyway, so the main idea was that you can just basically select the top ones and probably use some other traits as like markers and helpers. Actually, it also comes from research doing like in an adjacent lab with evolutionary algorithms. So they have some algorithms for like multi-objective optimization that has or like multi-criteria that have, and actually again coming back to this problem of generating hard tests. So they actually, while they optimize for one thing, like live, well, not there, it's not live, but runtime of the problem, they use several other markers like loop counters and so on to like optimize for this. So here the idea comes from this, so that you want to optimize one thing, but you can use other criteria to do this. So some kind of like genetic algorithm style solution were expected, or like manual, like selecting the top ones and crossing them.
Right. So for cows, you could just select based on their output, but for bulls, you look at the cows, you correlate the output with some other traits, and that's how you select the bulls. Is that the rough idea?
Like
and
for example, for basically the bulls have some genotype that are maybe increases the milk production or decreases. So you can't know it from their like phenotype, because they don't produce milk. But you can look at other traits and to say that okay, maybe ear size correlate with the milk production or something like this. I believe in the tests there like at least some traits are correlating with this. So maybe not 100%, but there is some correlation. But basically, yes, this is one of the problems that we don't know the solution, and we are eager to know how people did approach it.
Yeah, maybe it's more suitable for the qualification round when you had plenty of time and you could play with that.
Well, probably. I know. So one of the things we tried to have an interactive problem because it's makes the like round more fun, I would say.
Yeah, it's fun.
Like, for most of the people, it's it's fun to select some codes and to like look what happens and so on. So it's actually not many points are actually achievable by this problem.
Yeah, I think people didn't do very well on that problem, right?
Yeah.
Like even of the allocated points, not many were claimed.
Yeah, because we over-allocated basically points. So 100% actually almost the best call possible in this genome model. It's probably not reachable from the input.
That makes sense.
And in a reasonable time.
Yeah. Also, what I found interesting about this problem is that it sort of comes closest to the statistics machine learning field So you mentioned that some kind of clustering algorithms could be used in the previous one, in the reclustering. But this one sounds really like a problem for some machine learning algorithm to like figure out the features.
Yeah, so where you have some features, like there are several features that you can try to correlate to use them to basically say how good is the bull. So basically this problem is about bull selection at not cows, because for cows it's pretty straightforward, almost. But for bulls is harder.
Was there any penalty of just trying many options? Because you selected the maximum out of all possible like cows that I got back. Was there any limitations? Like if I sat all day and submitted all the options, would I be able to do like at least somewhat well?
Well, maybe depends on the. So there's some note in the problem statement that the results, like responses, not deterministic.
Yeah.
So basically, the breeding is non-deterministic. So actually, some of the participants maybe were more lucky than the others in what they get in the first round. So I know. So it was also like discussion points, like whether it's like the same for all of them, for all the participants, or random. So we decided, well, we don't want unlucky participants at the top.
Yeah. It would be better. This is my request for the next year. Do you, by the way, plan the Bioinformatics contest 2019?
Well, likely. So
please do.
Yeah. So we are like looking for problems for like sponsors and so on. So it depends on, and also, so the first contest was mainly just for fun for us, at least like for our ITMO team. So and the second one we try to also make it. So here in the ITMO, we open a master program in bioinformatics. So, and this is one of the ways to attract, and actually it's international. So it's one of the ways for us to attract the future students. So we'll see how it works. So if it works, we definitely will repeat it again. And also if we'll have a problem set, we also repeat it. And if we find some sponsors, we also repeat this.
Yeah.
So there are a couple of ways that we are going to do it again. And it's still fun, so...
Yeah, I have to say that regarding your advertising for your master's program, you didn't do a good job because I had no idea about the program. Like I participated, and this is the first time I hear about it.
Yeah, we will say about it in their final letter for the participants. So you will get the message about this.
Next time, put it somewhere in big letters. So I was saying that my request for the next time, if you do a problem like this, it would be nice if you could interact with the server, not just via this form on the website, but via some kind of API, like a HTTP request response, so that I could write a program that would automatically, of course, with some rate capping, but it could make the decision what to breed, sort of talk to the server, get the results back, analyze them all in this automatic mode. It will be, I think, much better than having an actual human person copy-paste the text.
Yeah, maybe. Even in the previous contest, we had also interactive problem. So when we tried to design it, how people will solve it and how they submit the solutions. So usually, in programming contests, you just submit a program that interacts with the checker.
Yeah.
However, here we have more broader audience, so they're not familiar with this, so and it can be complicated. So we decided this in this format, it's at least good for everyone. So you can just whatever the way you came up with the answer, you can submit it and then ask questions, so on. So we'll try to make other schemes, but yeah.
Yeah, I'm just saying you could keep this scheme where if someone doesn't know how to submit requests, they can just copy paste the text. But it's also nice to have a additional option of having a program just talk to the server itself.
Yeah, we will think about it. Yeah.
Cool. Well, guys, we've been chatting for quite a while. Thank you so much for your time. And on behalf of all the participants, thank you for organizing this. I look forward to the contest in the next year.
We hope for you to participate in it too, and probably the several others after that.
Thank you for inviting us. And we also want to thank our participants for taking their time and competing in the contest. And I also wanted to add that we're really thankful to the STEPIC team for their support on accommodating us.
And also, we'll send a feedback form soon. So any problems, ideas for problems, suggestions, and whatever other comments, just put it there. They will be very welcome.